Hello everyone, I'm Grégoire Brun from Université Catholique de Louvain in Belgium, and I will present the research that's made here at UC Louvain on electrochemical characterization of nitrocellulose membranes towards bacterial detection in water. Frequent water quality assessment and diligent water management are crucial to control water pollution and provide access to safe and sufficient water to human communities. Regarding drinking water quality, bacterial infections remain a major cause of illness and death, mainly in developing countries. Among the pathogens, Escherichia coli is of major interest. Moreover, the recent sanitary pandemic highlighted the need for mass screening the virus present in water environment, which is a method named wastewater-based epidemiology. This is indeed an effective approach to predict the potential spread of the infection in the population. In both cases, it is necessary to obtain water quality results in remote situations, at the place of diagnosis, and within a reasonable period of time. Current methods for the detection and quantification of bacteria, such as plate culture and bacterial counts, are very accurate but far too time-consuming, expensive, and require bulk laboratory equipment. This makes it absolutely unsuitable for rapid field analysis. There is therefore a real need for fast, portable sensors for point-of-care detection. And I will now quickly review some of the main point-of-care sensors used for water quality monitoring. In recent years, diagnostic tests in the form of paper strips, which are also called lateral flow assays, have emerged for application in medicine, food, and the environment, just like the well-known pregnancy test. These strips are based on the detection of pathogens by immunochromatography. The sample to be tested is deposited on a nitrocellulose membrane, which is quite a derivative of paper, and is guided to the test line by natural capillarity. At this point, the bacteria are immobilized by means of specific bioreceptors. The more usual method to detect the presence of the pathogens uses gold nanoparticles, which are specifically attached to the pathogens and emit red light in the presence of the pathogens, just as is shown on the slide. Several advantages of the paper substrates make this platform an ideal tool for field diagnosis of bacteria. First, it allows easy attachment of bioreceptors to provide immobilization for pathogens inside the membrane. Second, it has very interesting spontaneous microfluidic properties through capillarity. Third, it is low cost and enables easy manufacturing. And finally, it allows for volumetric detection methods, as the membrane could act as a filter and retain the bacteria from the whole water sample. The major drawback of such tests is the qualitative results that are obtained. Electrical measurements, on the other hand, enabled quantitative and precise biosensing. Common electrical measurements consist in detecting changes in the properties of materials close to electrode pairs when the targeted bioelements are immobilized. So the electric measurements offer a wide diversity for the characterization of materials and physical phenomena. Indeed, by accessing to the entire frequency spectrum, as can be seen on the left graph, this allows to explore much of the physical phenomena that are implied in biosensing applications. Moreover, the possibility to integrate electrical sensors with remote control systems, so just like smartphones, for example, really paves the way for very rapid and on-field point-of-care detections, including in developing countries. In order to get most of the advantages mentioned for both paper-based and electrical detection, in this work we propose to introduce and investigate how electrical measurement can be considered as a new means of detecting bacteria on the nitrocellulose used for the usual paper-based sensors. In particular, we study how to combine paper and electronic detection by introducing 4 innovations. First, we propose a phage endolysine, which was recently discovered by our microbiologist collaborators, as a new specific bioreceptor to specifically immobilize Bacillus thuringiensis wall cells on the nitrocellulose membranes. Bacillus thuringiensis are used here as model microorganisms. Second, we studied the developed biointerface to ensure that the bacteria are well captured over the whole membrane volume. Third, we propose to accommodate a common system for the dielectric characterization of materials to monitor here the change caused by the presence of bacterial cells in the membrane, and therefore to act as a biosensor. This setup consists in applying 2 parallel electrodes on both sides of the membrane. And finally, we fabricate an unconventional fringing field electrode setup, which are called interdigital electrodes, or IDE, to monitor the impedance change caused by the presence of bacteria inside the membrane, once again. This setup is used to validate the result obtained with the parallel placed setup. Let's now introduce the results section of this presentation by looking at the new bioreceptor chosen. The endolysine, which are produced by phage to lyse their host, present different domains with different purposes. In particular, the cell wall binding domain, quickly named CBD, is responsible to bind in close proximity to bacterial cells. This binding capacity is here used to use the CBD as a specific bioreceptor to immobilize and detect Bacillus bacteria wall cells. As can be seen on the right of the slide, we developed a simple deposition protocol to ensure the biofunctionalization of the whole membrane volume with the endolysine cell wall binding domains, then allowing for specific capture of the bacteria inside the pores of the membrane. In order to validate the protocol and the capture of the bacteria in the membrane, the proteins and bacteria were tagged for fluorescent microscopy. So using the fluorescence, we image the cross-section of the functionalized membranes. The image of the cross-section on the left shows that proteins were deposited on the whole membrane thickness, and we validated that bacteria are captured inside the membrane by looking into the pore using a confocal laser scanning microscopy. So on the right picture, you can clearly see that the bacteria, so in blue, are attached to the surface of the pore where the specific proteins are deposited. Since we know that the bacteria are specifically immobilized in the membrane, let's try to detect their presence by analyzing their electrical footprint. The parallel plate electrodes are commonly used to extract the dielectric properties of dry materials. In order to detect the presence of bacteria in the membranes, in this project we explore possibilities to measure the permittivity of a nitrocellulose membrane, which is filled with a buffer solution containing bacteria. So, as can be seen on the left figure, the setup consists of 2 electrodes between which the nitrocellulose membrane is inserted. Through impedance spectroscopy, these electrodes sense the electrical response of the sample over a frequency range between 1 kHz and 1 MHz. A software is used to extract the values for the permittivity at every frequency point. When bacteria are present in the volume of the nitrocellulose, its electrical properties are altered, and this enables us to observe a difference in permittivity with and without bacteria. A polyester backing layer is attached to the nitrocellulose membrane for ease of handling and to reduce the fragility of nitrocellulose. And this backing layer prevents currents to flow from one electrode to the other. So it is isolating nature, therefore screens the conductivity of the ionic solution in the nitrocellulose membrane, which makes it impossible to extract accurate conductivity values. Therefore, in the rest of this presentation, we focus on permittivity aspects since the measurements were the most coherent and repeatable. In order to perform the measurements, pieces of nitrocellulose membranes were dipped in two kinds of solutions. First, a blank physiological solution that was diluted in deionized water, and second, in the same solution to which 10 to the 8 bacteria colony counting units per milliliter were added. On this graph, the permittivity measured over the whole frequency range is represented for both the case with and without bacteria. So we observe a clear shift and a maximum sensitivity between 10 kHz and 100 kHz. For these measurements, we identified different sources of variability. A first parameter is the inhomogeneity of the membrane. We know that the nitrocellulose membrane presents a foam-like structure, and its structural anisotropy and the surface inhomogeneity render absolute measure of the permittivity really difficult, as the measurement setup is expected to hold homogeneous materials. A second source of variability is linked to the biofunctionalization protocol, which can sensibly modify the permittivity of the nitrocellulose sheet by altering its surface properties, for example, by developing a significant static charge on its surface. This indeed affects the dielectric measurements. Despite these variabilities, we succeeded in detecting the same trend between sample with and without bacteria. So relative measurements are thus possible between different samples. To explain this recurrent trend, we also tested membranes filled with saline solution of different ion concentrations. Qualitatively, you can see on the right graph that an increase in ion concentration induces the same relative change compared to the reference measurement than the bacterial solution compared to the blank solution, which is on the left-hand side. This strongly suggests that the bacteria are detected on the basis of an increase in ions due to the presence of the bacteria. This hypothesis is confirmed by another test we performed. We increased the ionic concentration of the physiological buffer in which the bacteria are suspended. So this leads to a decrease in sensitivity. Just you can see on the right graph. Even though the trends stay the same, the shift between the permittivities on the right graph is reduced by 20 to 40%, depending on the frequency, compared to the left graph. So this would mean that ions expulsed by bacterial cells are less contributing to dielectric changes in highly concentrated buffers than in lower ones. To validate the results we obtained with the parallel plates, impedance measurements were made with a periodic electrode design that is very common for sensing application, that has interdigital electrodes. So, as illustrated on the right graph, they offer the advantage that only a single-sided access to the nitrocellulose is required. Since these electrodes are planar fringing field electrodes, they are mostly sensitive to electrical phenomena that occur near the electrode surface plane. So we deposited gold interdigital electrodes directly on paper by means of an electron gun. Just like the left picture illustrates, a connection was made with an LCR meter through toothless crocodile clips. The solution, with or without bacteria, is directly applied on the paper surface with a micropipette. As explained, the goal is to validate the parallel plate measured with the interdigital electrodes. The coherence between the outcomes obtained with the parallel plates and the interdigital setups is verified via their equivalent electrical model. We do this by extracting the permittivity of the nitrocellulose membranes from the parallel plate measurement using their electrical model. And this value is then injected into the interdigital model to predict the values of the double layer capacitance and the capacitance of the nitrocellulose volume. Then we fit the complex impedance results from the interdigital measurements on the interdigital equivalent circuit, and this is done for nitrocellulose sample either with and without bacteria. And finally, an average increase of capacitance is observed for both the interdigit measurements and the predictions based on the parallel plate measurement. So, as just said, the presence of bacteria induces a variation of the double layer capacitance and the capacitance of the nitrocellulose volume, as seen by the interdigits. So this table here shows this variation as predicted based on the parallel plate measurement and as deduced from the interdigit measurement. The differential values have the same order of magnitude. We can then cautiously conclude that the main elements affected by the presence of bacteria are the modeled phenomena, that is the double layer capacitance, and the volumic electrical properties of the nitrocellulose. Furthermore, we show here that these elements are correctly considered in the equivalent electrical circuit. To conclude this presentation, we summarize now the main achievements of this work. First of all, an endolysin targeting Bacillus thuringiensis was successfully proposed as an innovative bioreceptor to capture whole bacterial cells in the nitrocellulose membranes. Then, we validated the use of parallel plates to characterize the dielectric properties of the membrane, with the aim of detecting bacteria in it. Two main parameters here influence the quality of the electrical measures. First, the biofunctionalization process generates uncertainty and variability in the measurement. Then, as the detection setup is mainly sensitive to ions, the initial buffer ionic concentration must be controlled to ensure robustness in the result. Then, we successfully demonstrated a proof of concept of bacteria detection within 5 minutes in nitrocellulose membranes by detecting bacillus cells in low salinity buffers. Our analysis suggests that the detection principle relies on the detection of additional ions that results from bacteria presence. However, deeper studies are required to understand the precise sensing mechanism and to gain more insights in the characteristic of ion released by bacteria and that contributes to the detection. Finally, this study opens the door for further research path on electrical bacteria sensing on nitrocellulose membrane. The addition of specific conjugated nanoparticles to this system shows hypermesis as the custom choice of nanoparticle would allow to better control the sensing principle, allowing for signal enhancement and then higher performances of the system. This will be the subject of future work. Thank you very much for watching this video. Ciao!